Good morning, everyone. I would like first to thank PacBio, KTest, and all the organization team for inviting me to this very exciting event. Today, I'm honored to share with you our research project on shotgun metagenomic study of gut microbiome from healthy Vietnamese adults. And this project aims to provide insights into the structure, the function, and health relevance of Southeast Asian populations, which has been so far underrepresented in the global microbiome databases. A microbiome, by definition, is a collection of genomes from all microorganisms living in a certain environment, which can be everything from a coral reef, a piece of arable land, to a shrimp gut. And these microbes grow, reproduce, and interact with each other and with the environment. So to have insights into the compositional and functional aspects of these microbes, of this microbial community. In other words, to know who they are and what they are doing is fundamental to microbiome study. And the methods for microbiome study include culture-based and unculture-based methods. The culture-based methods has been used for decades and have successfully identified numerous microbial species. But they do have limitations. They are inefficient regarding unculturable species, bacterial species, and they cannot truly reflect the interactions of these microbes in natural conditions. And on the other hand, unculture-based methods, especially the high throughput DNA sequencing technology, is independent of culturing. And for this technology underlies the metagenomics, which is the science that study the genomes of a microbial community. And there are two approaches for metagenomics. The first one is amplicon or targeted sequencing, and the second one is shotgun metagenomics. In the first approach, targeted sequencing, a marker gene or a few marker gene characteristic to a group of organisms or sequenced or amplified before sequencing. The second approach, the shotgun metagenomics, sequences the whole genome of all organisms obtained from this environment. So shotgun metagenomics provide more accurate information about the compositional and functional prediction, much more than the targeted sequencing technology. In this research project, we use shotgun metagenomics. So since birth, our body was colonized by numerous microbes. They include bacteria, archaea, fungi, viruses, with bacteria as the most dominant. So depending on the part of the body where they live, they form the oral, respiratory, skin, vagina, or gut microbiome. And among all of these human microbiomes, the gut microbiome is most important in terms of cell number, a little bit more than, nearly 40 trillions of cells, a little bit more than our 30 trillion cells, human cells. It is important Also in terms of diversity, from 300 to 500 bacterial species per person. And what is remarkable is that it encodes 150 more genes than our own cells. So this I think is why the gut microbiome, the human gut microbiome, is also called our hidden organ or our last organ. In healthy conditions, gut microbiome is stable but not static. It changes within an individual over time. It changes between individuals and between populations. And the changes are induced by internal and external factors. Internal factors such as the host genetics, age, gender, sex, or the immune status. And the external factors can be cited as diet, lifestyle, geography, medications, or environmental pollutions, among others. Diet is considered as one of the most important, if not the most important, impactful, not only important, impactful factors. We can take some examples. Studies have shown that a Western diet rich in protein, fat, and sugar promote Bacteroides dominant microbiome, whereas an Asian diet rich in fiber favor Prevotella or Segatella dominant microbiome. The main bacterial phyla in the gut microbiome include Bacteroides, Firmicutes, Actinobacteria and Proteobacteria, and a few others. Firmicutes and Bacteroides make up nearly 90% of the total, but this ratio change depending on the communities. The key functions of the human gut microbiome include digesting dietary fibers to extract energy and nutrients, to protecting our body against pathogens, producing vitamins, modulating our immune response, and even modulating our brain. What is very interesting is that any two individuals share about 40% of microbial taxa, while having in common nearly 90% of the functional metabolic pathways. And this is due to what we call the functional redundancy. That means that different species can perform the same functions within a community. So, since it performs so many functions, vital functions, important for our survival, our gut microbiome is deeply relevant to our health and our diseases. So, how does look like a healthy gut microbiome? Three characteristics for a healthy gut. Firstly is diversity, high number of taxa present. Secondly is resilience, a capacity of recovery after perturbations, and resistance to pathogens. And these qualities are assumed by a well-balanced microbial community. That means that any disturbance to this balance induce what is called dysbiosis and results in diseases, a large panel of diseases from metabolic conditions like type 2 diabetes, obesity, gastrointestinal disorders such as IBD, colorectal cancer, autoimmune or neurological disorders. So our research project on the gut microbiome of healthy Vietnamese adults involve 300 Vietnamese healthy people from Ho Chi Minh City, Vietnam, aged 20 to 60. Each participant provides 2 stool samples 1 month apart, and the DNA was extracted and sequenced in a short-read platform. The analysis included taxonomic profiling, the core microbiome definition, the classification of samples into enterotype, functional analysis, and furthermore, we use health indicator, the Gut Microbiome Wellness Index, to assess the health status of our cohort. This TACBA plot displays all of the 10 dominant species across all of our samples. Interestingly, is this bar, this dark blue bar, represents Prevotella cupri, a species that is very common to non-westernized people and is associated with fiber-consuming people. And in some sample, you can see that the abundance of this species is very, very high. So one question we ask ourselves is that what do we have in common with other populations in different geographic regions, and what makes us different from them? And we think that the 2 concepts that can answer to these questions are the core microbiome and the enterotypes. The core microbiome is a set of taxa clustered around a dominant genus and identifiable by this dominant genus. And is consistently present in all populations, several populations in uncertain environments. So, despite very high inter-individual variations in our samples, we were able to identify a core species which were found in more than 80% of our samples, with an abundance of at least 0.01%. At the right, you have the species components of the microbiome from different cohorts, from China, USA, India, Madagascar, Great Britain, the Netherlands. And in the left, you can see our 14 core species. These core species with this relative abundance have a clear overlap with the species found in other cohorts, indicated by the arrows. And the only species that was not found in other cohorts listed here is Mediterranean navus, which is a species commonly found in Western populations. So we can see that the core microbiome is specific to a community. So is there any defined combination of bacteria that is more universal? In 2011, Pierre Bock and his associates mentioned the notion of enterotype. And according to them, enterotypes are a set of microorganisms identifiable by one dominant taxa. They co-occur but is identifiable through this dominant genus. And since their discovery, studies all over the world reported enterotypes, different enterotypes. The most known are Bacteroides, Prevotella, and Ruminococcus or Blautia. But there are differences. In this slide, you can see that in a cohort, there are only two enterotypes, the Prevotella and the Bacteroides enterotypes, whereas in the Chinese cohort, they reported 5 enterotypes. In our cohort, we can classify our samples into 3 enterotypes, the Bacteroides, the Segatella, and the Blautia enterotypes. This PCOA plot with Bray-Curtis similarity shows very clear clustering of samples into the 3 enterotypes. So for functional analysis, this heat map shows the 30 most dominant pathways found in the 3 enterotypes. The horizontal upper bar here represents the 3 enterotypes with different colors, and the vertical bar corresponds to different metabolic activities, carbohydrate, nucleotide, and lipid metabolisms with other functions. And in this heat map, you can see patterns that are specific to one enterotype. For example, this one, very specific to Blautia enterotype, or this pathway that was shared by Bacteroides and Blautia. And Prevotella, and Segatella. To look more into the dominant metabolic pathways that are specific to each enterotype, we use left-C analysis. And the analysis this is the result that shows the 10 most dominant pathways specific to each enterotype. For the Bacteroides enterotype, the amino acid, cofactor and vitamin metabolism, the nucleotide and lipid metabolism suggests diet relatively poor in nutrients and poor in fiber. For Blautia enterotype, sugar and carbohydrate metabolism, together with cofactor and vitamin biosynthesis and nucleotide degradation, suggests a fermentative microbiome that can use efficiently dietary sugar. And in the last enterotype, the Segatella enterotype, the predominance of energy, polyamine, and NAD metabolism together with nucleotide degradation pathways suggests an adaptation to stress. So since our health, depends so much on our gut microbiome, it's quite tempting to try to assess the health of an individual based on his or her gut microbiome. And that is the objective of the Gut Microbiome Wellness Index, GMWI, version 2. This GMWI 2 is a machine learning based classifier that was trained in over 8,000 metagenomes coming from 26 countries and spanning 6 continents. and it covered healthy and people people with 11 different diseases. The GMWI can distinguish with an accuracy of nearly 80%. A healthy people from a non-healthy people. Healthy people will have a positive GMWI score, whereas the non-healthy will have a negative GMWI score. So we apply this health indicator to assess the health status of our cohort for two purposes. The first one is to know or to confirm if our cohort is really a healthy cohort and how it is situated among all healthy cohorts in other regions. And the second purpose is to assess the ability of using the GMWI as a health indicator for Vietnamese populations. In this box plot, you can see that all of these are GMWI values calculated from different cohorts coming from Europe, USA, China, and other countries like Japan, India, Madagascar, Israel, and others. And all of these represent the healthy populations from these study cohorts. And you can see that our cohort, framed in red here, has values, GMWI values falling in the range of the global healthy range. That means, firstly, that our population is in fact representative to a healthy population. And secondly, that the GMWI is quite a robust, transferable model that can be applied in Vietnamese populations with adjustments. In summary, I would say that we have identified a core microbiome of 14 species from 33 individuals who are healthy Vietnamese people, Segatella copri and Mediterranibacter navus, some distinctive features that are different, that show difference between our cohort with others. We can classify our samples into 3 enterotypes: the Bacteroides, the Segatella, and Blautia enterotype. And for functional analysis, we identified 30 dominant pathways for the 3 enterotypes, with a few distinctive dominant pathways specific to each enterotype. And lastly, the application of GMWI in a healthy population in Vietnam to show that it can be used to assess the health status of our Vietnamese populations. What we would do next is that we would enlarge this presentation comes from the analysis of more than 30 people. Which we will do next is to enlarge the full cohort, and we hope that we will have refined findings. Nevertheless, I think that this work constitutes a foundational data set for microbiome research in Vietnam and contributes to the global efforts to map gut microbiome across populations. These are our team working on this work, and thank you for your attention.